The concept of a gene is a fundamental principle in genetics that explains how traits are inherited from parents to offspring. A gene is a segment of DNA that encodes a specific portion, protein, or RNA molecule, which in turn determines a particular trait or a phenotype. The study of genes and their inheritance is known as genetics. Alleles are alternative forms of a gene that exist at the same locus on a chromosome. Each individual inherits two alleles per each gene, one from each parent. If the two alleles are the same, the individual is homozygous for that gene. If the two alleles are different, the individual is heterozygous for that gene. Multiple alleles refer to the existence of more than two alleles at a given gene locus. Pseudoalleles are alleles that do not affect the phenotype of the individual but can be distinguished from other alleles using genetic tests. Complementation tests are used to determine whether two different mutations that result in the same phenotype affect the same gene or different gene. If the mutations affect the same gene, they will not complement each other and will produce the same phenotype when combined. If the mutations affect different genes, they will complement each other and produce a wild-type phenotype when combined. The importance of understanding the concept of genes, alleles, and complementation tests lies in the fact that it helps in the study of inheritance patterns and the transmission of traits from one generation to next. So, complementation tests are used to determine whether two different mutations that result in the same phenotype affect the same gene or different genes. If the mutations affect the same gene, they will not complement each other and will produce the same phenotype when combined. If the mutations affect different genes, they will complement each other and produce a wild-type phenotype when combined. And learning about these topics also helps to provide insight into genetic variation, genetic diseases, and the evolution of species. Then extension of Mendelian principles. And extensions of Mendelian principles: codominance, incomplete dominance, gene interactions, pleiotropy, and related topics. So, codominance. In this type of inheritance, both alleles are expressed equally in the phenotype of the heterozygote. An example of codominance is the ABO blood group system in humans, where both the A and B alleles are expressed in the AB blood group. Then, incomplete dominance. In this type of inheritance, the heterozygous phenotype is intermediate between the homozygous dominant and homozygous recessive phenotype. An example of incomplete dominance is the flower color of snapdragons, where the heterozygous offspring have a pink flower color that is intermediate between the red and white flower colors of the homozygous parents. Gene interactions. In gene interactions, multiple genes work together to produce a single phenotype. An example of gene interaction is the coat color of Labrador retrievers, where three genes work together to produce the different coat colors. Then pleiotropy. In pleiotropy, a single gene can affect multiple traits. An example of pleiotropy is sickle cell anemia, where a mutation in the beta globin gene causes red blood cells to be mishapen and also provides protection against malaria. Next, genomic imprinting. In genomic imprinting, the expression of a gene depends on whether it was inherited from the mother or the father. An example of genomic imprinting is Prader-Willi syndrome, where this syndrome only occurs when the genes responsible are inherited from father. Then, penetrance and expressivity. Penetrance refers to the proportion of individuals with a particular genotype that show the expected phenotype, where expressivity refers to the degree to which a particular phenotype is expressed in individuals with the same genotype. Phenocopy. The phenocopy, an environmental factor that produces a phenotype that is identical to a phenotype caused by a genetic mutation. An example of phenocopy is the effect of thalidomide on limb development in fetus, which produces a phenotype similar to the genetic condition phocomelia. Linkage and crossing over. Linkage refers to the tendency of genes on the same chromosomes to be inherited together, while crossing over is the process by which homologous chromosomes exchange genetic material during meiosis. Sex linkage. Sex linkage refers to the genes that are located on the sex chromosomes X and Y and are inherited differently in males and females. An example of sex linkage is color blindness, which is more common in males because the gene responsible is located on the X chromosome. Then sex-limited and sex-influenced characters. Sex-limited traits are expressed only in one sex, while sex-influenced traits are expressed differentially in males and females due to hormonal differences. An example is a limited trait is the growth of antlers in male deer, while an example of a sex-influenced trait is baldness, which is more common in males due to the effect of testosterone. Then gene mapping methods. Gene mapping refers to the process of determining the relative positions of genes on a chromosome and their distances from each other. Mapping the genes provides an understanding of the genome structure and helps in identifying genes responsible for specific traits. There are several methods available for gene mapping. Some of the most commonly used methods are linkage maps. Linkage maps are created based on the analysis of the frequency of recombination between genes. The closer two genes are each other on a chromosome, the less likely they are to be separated during meiosis, and the lower the frequency of recombination between them. Linkage maps can be used to determine the relative positions of genes on a chromosome and the distances between them. Then tetrad analysis. Tetrad analysis is a method used in fungi to determine the linkage between genes. It involves the examination of 4 haploid spores produced by a single meiotic event and analysis of the phenotypes of the spores to determine the linkage between the genes. Mapping with molecular markers. Molecular markers are specific DNA sequences that can be used to identify the location of genes on a chromosome. The most commonly used molecular markers are restriction fragment length polymorphisms, microsatellites, and single nucleotide polymorphisms, SNPs. By analyzing the inheritance patterns of these markers, the location of genes can be determined. Mapping by using somatic cell hybrids. Somatic cell hybrids are created by fusing a somatic cell with a cell that lacks a nucleus. The resulting hybrid cell contains 2 sets of chromosomes, one from each of its parent cell. By analyzing the hybrid cell, the location of the genes can be determined. Then development of mapping population in plants. In plants, mapping populations can be developed by crossing 2 varieties of plants that differ in a particular trait. By analyzing the inheritance patterns of the traits in the offspring, the location of the gene responsible for that trait can be determined. Next, extrachromosomal inheritance. Extrachromosomal inheritance refers to the inheritance of genetic material that is not present on the chromosome. Mitochondrial and chloroplast genes are responsible for extrachromosomal inheritance as they are located outside the nucleus in organelles such as mitochondria and chloroplasts. Then mitochondrial genes are inherited maternally, which means that they are passed on from the mother to her offspring. This is because the egg cell contributes most of the cytoplasm, including the mitochondria, to the developing embryo. In contrast, the sperm cell contributes very little cytoplasm and only a few mitochondria, which is usually destroyed shortly after fertilization. Therefore, the offspring inherit most of their mitochondrial DNA from their mother. Chloroplast genes are also inherited maternally in most plants. This is because the egg cell contributes most of the cytoplasm, including the chloroplasts, to the developing embryo. However, in some plants, such as in liverwort mercantia, chloroplast genes are inherited paternally or biparentally. The inheritance of mitochondrial and chloroplast genes has important implications for the study of evolution and phylogenetics, as they can provide information on the ancestry and relationship of different organisms. They are also important for the study of genetic diseases, as mutations in mitochondrial genes can cause a variety of disorders. Next, microbial genetics. Microbial genetics is the study of the genetics of microorganisms, including bacteria, viruses, and fungi. Microbial genetics plays a vital role in understanding the mechanisms of genetic variation and gene regulation in microorganisms. So, methods of genetic transfer include transformation. Transformation is the process by which bacteria take up DNA from their surroundings and incorporate it into their genome. In bacteria, transformation occurs naturally in some species such as Streptococcus pneumoniae and can be induced induced artificially in others such as Escherichia coli. Then conjugation. Conjugation is a type of sexual reproduction in bacteria in which two cells physically connect to transfer genetic material, usually in the form of a plasmid. The plasmid contains genes that confer beneficial traits such as antibiotic resistance or the ability to degrade complex compounds. Then transduction. Transduction is the transfer of genetic material from one bacterium to another by means of a virus. During the process of transduction, a virus infects a bacterial cell, and during replication, it incorporates a piece of bacterial DNA into its own genome. When the virus infects a new bacterial cell, it transfers this piece of DNA, which can be integrated into the recipient bacterium's genome. Sextinction. Sextinction is a rare form of conjugation that occurs when a bacteriophage infects a bacterium carrying a conjugative plasmid. The bacteriophage carries the plasmid along with it when it infects a new bacterium, which can then acquire the plasmid by conjugation. Then mapping genes by interrupted mating. Interrupted mating is a technique used to map genes in bacteria. In this technique, bacterial cells are allowed to conjugate, and the mating is then interrupted at specific intervals by physical disruption of the cells. By analyzing the frequency of gene transfer by different time points, researchers can infer the location of genes on the bacterial chromosomes. Fine structure analysis of genes. Fine structure analysis of a gene is a technique used to map the precise location of genes on the chromosome. This technique involves the use of bacteriophages that can integrate into the bacterial chromosomes at specific sites. By analyzing the frequency of gene transfer at different points along the chromosome, researchers can create a detailed map of the location of genes. So, understanding microbial genetics is essential for developing new therapies and treatments for infectious diseases. By studying these genetic mechanisms by which microorganisms evolve and adapt to their environment, researchers can develop new strategies for preventing and treating bacterial infections. Additionally, microbial genetics is important for understanding the process of genetic recombination and mutation, which play a critical role in the evolution of all organisms. Then human genetics. Pedigree analysis is a tool used in human genetics to study the inheritance of traits within a family. It involves the examination of the relationships and characteristics of individuals across generations to determine patterns of inheritance. The patterns observed can be used to determine whether the trait is dominant or recessive and to estimate the probability of an individual carrying or inheriting the trait. The load score is a statistical measure used to assess whether two genetic loci are likely to be inherited together due to linkage. It is calculated by comparing the probability of observing the linkage between the two loci to the probability of observing the same linkage by chance. A load score of 3 or higher indicates strong evidence for linkage. Then karyotypes are images of an individual's chromosomes arranged in order from largest to smallest. They can be used to diagnose chromosomal abnormalities such as Down syndrome, Turner syndrome, and Klinefelter syndrome. Genetic disorders are conditions caused by changes or mutations in an individual's gene or chromosomes. Examples include cystic fibrosis, sickle cell anemia, Huntington's disease, and hemophilia. These disorders can be inherited in a variety of ways, including autosomal dominant, autosomal recessive, and X-linked inheritance. Understanding the genetics of these disorders can help in the diagnostic purpose and treatment of affected individuals, as well as in genetic counseling for families at risk of passing on the condition to their offspring. Then quantitative genetics, polygenic inheritance, heritability and its measurements, and related topics. So quantitative genetics is a study of the inheritance of quantitative traits, which are traits that are influenced by multiple genes and the environment. These traits are usually measured on the continuous scale, such as height, weight, or yield. In this field, researchers use statistical methods to study the genetic and environmental factors that influence the variation of these traits. Polygenic inheritance refers to the inheritance of traits that are controlled by multiple genes. Unlike single gene traits that follow Mendelian inheritance patterns, polygenic traits are influenced by the combined effects of many genes, each with a small effect on the phenotype. Examples of polygenic traits include height, skin color, and intelligence. Heritability is a measure of the portion of the phenotypic variation that is due to the genetic factors. It is expressed as values between 0 and 1, where a value of 0 indicates that all variation is due to environmental factors and a value of 1 indicates that all variation is due to genetic factors. Heritability estimates are important for understanding the genetic basis of complex traits and for predicting how traits will respond to selection and breeding programs. QTL mapping, or quantitative trait locus mapping, is a method used to identify regions of the genome that are associated with the variation of a quantitative trait. This method involves analyzing the genetic variation of a population and measuring the association between the genetic variation and the variation of the trait of interest. By identifying the genomic regions associated with the traits, researchers can gain insight into the genetic architecture of the trait and identify candidate genes that may be responsible for the observed variation. So overall, quantitative genetics is important for understanding the genetic basis of complex traits and for developing breeding strategies to improve these traits in crops and livestock. It also has applications in human health, where it can be used to study the genetic basis of complex diseases and to develop personalized medicine approaches. Then mutation and related topics. So mutation refers to a permanent alteration in the nucleotide sequence of DNA that can lead to changes in the phenotype of an organism. So mutations can occur naturally or can be induced by exposure to mutagenic agents such as radiation, chemicals, or viruses. Mutations can have a wide range of effects, from being lethal to being silent or neutral to producing beneficial or harmful traits. So types of mutations. Point mutations. These are the most common type of mutations that involve a change in the single nucleotide base pair. Point mutations can be of two types, like substitution. In this type of mutation, a single nucleotide is replaced by another nucleotide. Then insertion or deletion. In this type of mutation, one or more nucleotides are added or deleted from the DNA sequence. Then chromosomal mutations. These mutations involve changes in the structure or a number of chromosomes. Chromosomal mutations can be of four types, like deletion, loss of a segment of a chromosome; duplication, a segment of a chromosome is repeated; inversion, a segment of a chromosome is reversed in orientation; then translocation, a segment of one chromosome is transferred to another chromosome. Then genome level mutations. These mutations involve changes in the number of chromosomes, such as polyploidy. Causes of mutations include spontaneous errors during DNA replication or repair, exposure to mutagenic agents such as radiation, chemicals, or viruses, errors during mitosis or meiosis. Then how to detect mutations. Detection of mutations can be detected using various techniques, including DNA sequencing. This technique allows the determination of the exact nucleotide sequence of a DNA molecule. Polymerase chain reaction (PCR). This technique allows the amplification of specific DNA sequences, making it easier to detect mutations. Then gel electrophoresis. This technique separates DNA molecules based on size, allowing for the detection of mutations that cause changes in the size of a DNA fragment. Then important mutant types. Lethal mutants. These mutants cause death of the organism either during embryonic development or after birth. Then conditional mutants. These mutants exhibit a phenotype only after specific conditions, such as change in temperature or presence of a particular chemical. Biochemical mutants. These mutants have a specific defect in a biochemical pathway, leading to the accumulation or deficiency of a particular metabolite. Loss of function mutants. These mutants result in the loss of function of a particular gene or protein. Gain of function mutants. These mutants result in the gain of a new function or activity of a particular gene or protein. Importance of mutations are important for following reasons, like they provide genetic variation upon which natural selection can act, leading to adaptation and evolution. They can be used to study gene function and regulation. They can be used to develop new crop varieties, improve animal breeding, and treat genetic diseases. Then mutation types, causes, and detection. Mutation is a permanent alteration in the DNA sequence that makes up a gene. It is a natural process that creates genetic variation, which is essential for evolution. Mutations can be caused by a variety of factors, including errors in DNA replication, exposure to radiation, chemicals, and viruses. There are different types of mutations, like point mutations, a change in a single nucleotide in the DNA sequence. There are three types of point mutations: silent, missense, and nonsense mutations. Then frame shift mutations, a deletion or insertion of one or more nucleotides that changes the reading frame of the gene. This type of mutation can have significant consequences on the resulting protein. Then chromosomal mutation, changes that affect the structure or number of chromosomes. Examples include deletions, duplications, inversions, and translocations. Then causes of mutations, spontaneous mutations, random errors that occur during DNA replication. Then mutagens, physical or chemical agents that increase the frequency of mutation. Examples include radiation, chemicals, and certain viruses. Then detection of mutations, DNA sequencing. The most accurate way to detect mutation is by sequencing the DNA. Then polymerase chain reaction is a technique used to amplify specific regions of the DNA for analysis. Then mutant types, lethal mutation, conditional mutation. Conditional in the sense a mutation that has an effect only under certain conditions, such as temperature or presence of a particular chemical. Then biochemical mutation, a mutation that affects the function of a protein. Then loss of function mutation, a mutation that results in the complete or partial loss of a normal function of a gene or protein. Gain of function mutation, a mutation that results in a new or enhanced function of a gene or protein. Then germinal mutation, a mutation that occurs in a germ cells and is passed on to the next generation. Then somatic mutation, a mutation that occurs in the body cells and is not passed on to the next generation. Insertional mutagenesis, a technique used to induce mutations by inserting a foreign DNA sequence into the genome. Importance of mutations, evolution. Mutations are the source of genetic variation which is essential for evolution. Then diseases. Mutations can cause genetic diseases such as cancer, cystic fibrosis, and sickle cell anemia. Next, biotechnology. Mutations can be used to produce new strains of crops of microorganisms with desirable traits. Structural and numerical alterations of chromosomes, deletion, duplication, inversion, translocation, ploidy, and their genetic implications. Structural and numerical alterations of chromosomes can lead to significant genetic implications. Deletion. Deletion occurs when a segment of chromosome is lost during the process of cell division. The lost segment may include one or more genes, and the severity of the genetic effect depends on the size and location of the deletion. Deletions can lead to genetic disorders such as the Cry-du-Chat syndrome, which results from a deletion on chromosome 5. Then duplication. Duplication occurs when a segment of chromosome is copied and added to the same or another chromosome. This can result in the presence of extra genetic material which can have a significant impact on an individual's health. Duplication of the 21st chromosome is responsible for the development of Down syndrome. Then inversion. Inversion occurs when a segment of chromosome is flipped in orientation. This can result in the reversal of the order of genes within the segment, which can disrupt the normal function of these genes. Inversions can be inherited from parents or can arise spontaneously during cell division. Next, translocation. Translocation occurs when a segment of one chromosome breaks off and attaches to another chromosome. This can result in the fusion of genes from two different chromosomes, which can disrupt normal gene function. Some translocations are associated with genetic disorders such as chronic myelogenous leukemia. Then ploidy. Ploidy refers to the number of sets of chromosomes in a cell. Humans are normally diploid, meaning they have two sets of chromosomes. However, some individuals may have extra sets of chromosomes, triploid, tetraploid, etc., or missing sets of chromosomes like monosomy, nullisomy, etc. These conditions can have significant genetic implications and can result in developmental disorders or miscarriage. So recombination, next topic, recombination and related topics. Recombination is a genetic process in which new combinations of alleles are produced by the exchange of genetic material between chromosomes or within a chromosome. So homologous recombination involves the exchange of genetic material between two chromosomes or between two regions of the same chromosome that share similar DNA sequences. This process is important for the repair of damaged DNA, the criterion of genetic diversity during meiosis, and the generation of new recombinations of alleles in populations. Non-homologous recombination, also known as non-homologous end joining, involves the joining of two broken ends of DNA that are not similar in sequence. This process is involved in DNA repair and can result in the creation of genetic mutations. Transposition is another form of non-homologous recombination in which mobile genetic element such as a transposon moves from one location in the genome to another. Transposition can result in genetic rearrangements and mutations and can also be important for the evolution of new genes and the adaptation of organisms to changing environment. Recombination has important implications in genetics, including the generation of genetic diversity, the evolution of new traits, and the occurrence of genetic diseases. Recombination can also be used in genetic engineering to create new genetic recombinations and to introduce new genes into organisms. Next major topic, diversity of life forms. Principles and methods of taxonomy in detail. Taxonomy is the science of identifying, classifying, and naming living organisms based on their morphological, physiological, and genetic characteristics. The principles and methods of taxonomy are important for understanding the diversity of life forms and how they are related to each other. The principles of taxonomy include the use of homologous features to group organisms, hierarchy classification, and the use of binomial nomenclature. Homologous features are traits that are similar between different species due to shared ancestry. These traits can be used to group organisms into related taxa such as families, orders, and classes. Hierarchical classification involves organizing this taxa into a system of nested groups, with the most inclusive group being the kingdom and the most specific group being the species. Binomial nomenclature involves giving each species a unique two-part scientific name consisting of its genus and species names. For example, the scientific name for humans is Homo sapiens, where Homo is the genus name and sapiens is the species name. The methods of taxonomy involve the collection of data about organisms, the analysis of that data, and the use of that analysis to classify organisms. These methods include the use of morphological, physiological, and genetic data. Morphological data involves examining the physical characteristics of organisms such as their shape, size, and color. Physiological data involves studying the functions and processes of organisms such as their metabolism and behavior. Genetic data involves analyzing the DNA and RNA of organisms to determine their genetic relationships. One of the major tools used in taxonomy is the creation of a taxonomic key. This is a tool that allows scientists to identify unknown organisms based on their physical characteristics. Taxonomic keys consist of a series of questions or statements that lead the user to the correct identification. For example, a key for identifying birds might ask whether the bird has a hooked beak or a straight beak and whether it has webbed feet or not. Next topic we will be learning tomorrow. Thank you for listening.